Hi everyone, how's it going?
Hi, olá a todos. Muito obrigado a todos os presentes em assistir uma vez mais um dos CEBIMários, o CEBIMar. Esta vez a gente terá o prazer de assistir o Dr. Chance McDonald, que vai falar um pouco sobre peixes em ambientes profundos. E eu quero, com esse último CEBIMário, fechar o ano. Talvez muitos de vocês já estão de férias e agradeço que esteja ainda aqui assistindo o nosso webinário. Então, eu vou apresentar o Chance e passar a palavra para ele. Então, thanks, Chance, for accepting your talk today. It's very important for the webinar, this presentation, because it is one for internationalization. And, well, maybe you can talk more about you and the talking you is this room is yours in this moment. Thanks for
Okay,
accepting, and you have 30 minutes for your presentation. Thank you.
okay, no problem. Hey, thanks Juan, thanks for inviting me, and I'm really excited to be here today to talk with you all about some of the research that I've been doing. So yeah, my name's Chancey MacDonald. I did a PhD at James Cook University and the Centre for Excellence for Coral Reef Studies with Jeff Jones as my supervisor. I finished that a couple of years ago. I had a bit of a late start. And for the last 11 months, I've been at the California Academy of Sciences as a postdoctoral researcher with Luis Roca. And I've been doing a bit of work with Hudson Pinero, which some of you may know, and also with Juan. So the stuff that I'm going to be talking to you about today is about depth gradients and the pattern, process, and risk mitigation amongst coral reef fishes. So the first thing that I wanted to remind you all is that, oh, sorry, I skipped a slide here. First thing I wanted to remind you all is that most organisms are distributed along environmental gradients. And here we have a tropical plant community changing along an altitudinal gradient from a high energy, high competition environment at the low end of the altitudinal range through to lower density environments up towards the edge of their range margin, where competition is lower and resources are diminished. Another example of a classic environmental gradient is the change in sea surface temperature from the equator to the poles. And of course, we know that global environments are changing dramatically in the Anthropocene. And this has led to wide-scale habitat degradation and loss, shifts in habitat boundaries of many species, and contractions and expansions in the viable ranges, with examples available in most taxon ecosystems that have been studied to date. One of the most well-documented examples is of birds and butterflies in Northern Europe and the UK having northern range expansions due to heating temperatures. And so this has led to a renewed impetus to study the distributions of species along gradients, the ecological drivers of those distributions, and also the vulnerability of ecosystems and species going into the future. So to put that succinctly, a key ecological question in Anthropocene is how does ecological change interact with environmental gradients and species ecologies to influence their vulnerability? So if we go back to our altitudinal gradient and put a couple of frogs on here as a frog species and introduce some environmental change, whether it be from land use change or increasing temperatures, the species might be able to be resilient to that for a while. But if it continues too much, then some of the population might have to move further along the gradient. And of course, if environments continue to change, they have opportunities to move further along the gradients where they might find an ecologically viable refuge. But if some of those habitats aren't viable refuges, or if environmental changes too much and they try to move too far along the gradient, at some point it'll be too far and you'll have species attrition. So in order to understand ecological change and vulnerability along gradients, there's a few key things that you might want to know. The first of these is what are the natural distributions of species and their habitats along the gradients? Are species narrowly distributed along a gradient? Are they broadly distributed? their distributions constrained by the biotic habitats that they're associated with. The second thing you might want to know is something about the relationships between distributions and vulnerability traits. For example, species that are resource specialists are generally considered to be less abundant and less widespread in their environment, and therefore more vulnerable than generalist species. You also want to understand something about the variation in space use and effort required to secure the appropriate amount of resources. For example, at the range margins, there might be fewer resources, which will result in longer searches for foods, which would be higher costs for species living at the range margins. But on the other hand, you might have other ecological variables in play, like, for example, there might be less competition towards the range margin. You might also want to know something about changes in behavior along environmental gradients. So species that can show behavioral plasticity in their diet and habitat use, for example, might have decreased vulnerability as environments change, or as they occupy different positions along an environmental gradient. And the last thing you want to know is, actually, are there spatially differential impacts and outcomes of disturbance? So is there actual evidence of changing risk and vulnerability along environmental gradients? Now I work on coral reefs. You might think of that as an extension of the tropical gradient, moving down instead of up. And of course, coral reefs are in critical condition, or moving into a critical condition going into the future, because of warming climate and warming sea surface temperatures in particular. And this has led to global coral reef degradation, for example, from more water bleaching or storm damage. And we know that when we have reef degradation, we have widespread loss of fish abundance, quite often widespread changes in fish assemblages, and this is mostly because we have dramatic declines in coral-dependent specialists. For example, species that feed on corals or species that live within corals have the highest change in abundances per change in coral cover compared to other trait groups. But much of what we know about reef degradation, in fact, the ecology of coral reef fishes, is from the shallowest 10 or so meters on coral reefs. And of course, distributions of organisms are quite often a lot deeper than this. So here we're looking at the distribution of Acropora, a key habitat forming coral. And you can see that in the tropics, quite often get distributions down to 40 or 60 meters or more. And much of what we know, as I said, is in the shallowest 10 meters. And much of my research has been interested in trying to figure out what is going on in these deeper depths. And so that's what I did a number of years ago when I decided to jump in and start to research the ecology of these deeper reefs. So of course, depth has a lot of effects on coral reefs. You have a strong reduction in light and temperature, which results in a decline in coral cover, generally a decline in the number of species in corals and in fishes, not so much in things like sponges. And then also you get a decline in fish densities. But one other thing that we're starting to notice is that you quite often get a decline in disturbance effects, particularly bleaching. There's been a number of papers that have come out in the last couple of years that have showed some reasonably strong attenuation of bleaching effects as you move along the depth gradient. I have to say this isn't always the case, and it's probably not a panacea. But anyway, this has led to question whether there's potential for increased survival opportunities for the proportions of populations of species that live on the deeper reefs that might be vulnerable to changes in the ecosystem in shallow reefs. And so, for example, depth is a major factor in forming post-disturbance recovery trajectories, and is also a major influence on reef fish extinction threat predictions. Of course, Luis Roja and his team will remind you that deep reefs definitely aren't untouched, and that there's a large number of degradation events that do occur on the deep reefs as well as shallow reefs, and they need protecting in their own right. But basically, if we flip our mountain example upside down, and we ask the question whether deeper reefs might be able to provide a refuge because they might have a more stable habitat, put a couple of species on there. The question is if we have disturbance that is mostly limited to shallow waters, are there some species that will do better than others? Those that are species that are vulnerable, will they potentially be able to move down deeper along the gradient? And even if they can do that, will the cost be too high for them to be able to have sustainable populations? And in order to understand this, we need to first understand depth-related dynamics in their ecology. So my broad research aims over the past few years have been to understand the pattern and limiting processes of natural depth distributions in coral reef fishes, as well as to investigate the depth-related ecological changes that may aid or hinder their ability to succeed in deep reef environments. And I hypothesize that deep reefs will show potential to act as refuges if, first of all, many species have broad or deep depth distributions, if ecologically vulnerable species are not limited to shallow waters, if deep reef residents can efficiently secure resources, if depth-related diet changes do not incur sublethal costs, and if in fact the deeper reef assemblages are more resilient to coral loss. And for a lot of the work that I've done to date, and for a lot of work that I'll be presenting today, has been based in Kimby Bay in Papua New Guinea. It's on the edge of the Coral Triangle, just above Australia. It has a very steep bathymetry, and that means that we have very deep, clear water reefs close to the coast, which makes it an excellent place to study these kind of depth refuge hypotheses. So the first question is, do many species have broad or deep depth distributions? So just showing you some highlights here. To answer this question, within this study system in Kimby Bay in Papua New Guinea, I recorded the depth distributions or abundance distributions of 123 species from 4 reef fish families that represent a broad suite of trophic traits between 0 and 40 meters. And you can see that that 25% of the species were limited to the shallowest 5 meters. So these are species that might be vulnerable to changing environments going into the future. But 25% of species also had very broad depth distributions from 0 to 30 meters, and 12% of species had a broad depth distribution from 0 to 40 meters. Now, importantly and interestingly, 85% of the species that had strong coral associations had some representation at 20 meters or below. So it seems as though quite a number of species have broad depth distributions, at least within this depth gradient that I've been looking at. The second question is, are ecological vulnerable species limited to shallow waters? And of course, having 85% of strongly coral associated species at 20 meters or below goes some way towards answering this question, but I wanted to answer it in finer detail. And so I turned to the butterfly fishes, the Chaetodontidae family, as a model study group. And I'm sure Dave Bellwood would have you say that I did this because the fish are yellow. And that's partially the case. They're easy to see on the reef, so you can find out a lot about them because you can follow them around. But also, they're ecologically very interesting in that they have a broad range of specialization on corals. So you have obligate corallivores that only feed on corals, facultative corallivores that sometimes feed on corals, sometimes feed on invertebrates, invertebrate feeders that only feed on invertebrates, and then planktivores. And you can see that this broadly corresponds to some of the different response levels that we've seen to coral reef degradation in shallow waters in past events. So there's some expected relationships among depth range, abundance, and coral specialization. So we're asking, what are the species with broad depth ranges? What are their specialization levels? And are specialized species limited to shallow waters? And you can see here that the expectation is that species with broad depth ranges would be generalists, and highly specialized species would have narrow depth ranges. And likewise, specialized species would have low local abundances. And so you've got this kind of increased vulnerability here where you have small depth ranges and low local abundances. So if we look at the abundance distributions of the butterfly fish within Kimby Bay, you can see actually that our first expectation wasn't the case. The most specialized species were actually had the broadest depth ranges in the system. And then if we look at the local abundances, we can actually see that the species with the broadest depth ranges did have the highest local abundances, but instead of being generalist species, the species that were most specialized on corals had the highest local abundances. So they're actually sitting quite well in this system in terms of broad depth distributions, high abundances, despite being highly specialized on corals. So species with broad depth ranges are abundant and highly specialized. And if you look at a trait group level as well, the obligate corallivores here in the orange were more abundant than facultative and non-corallivores at all depths. Of course, it's not enough just to know the depth ranges. You need to know something about the abundance distributions of the organisms within these depth ranges. And so here you have 4 obligate corallivore species that all have depth ranges that extend from 0 to 40 meters, but they also have decreasing dietary specialization from left to right, with Chaetodon baronessa that feeds on very few coral genera and Chaetodon octofasciatus that feeds on many coral genera. And what you'd expect is that the more specialized species would have abundances that are biased towards shallow waters. And that is definitely what we saw. So the more specialized species have more of their abundance in shallow water, less of their abundance in deep water. And the more generalist species actually have very few individuals occupying the shallow waters, but higher abundance in deeper waters. So the next question we wanted to look at was, okay, you've got these potentially vulnerable coral specialists on the deep reefs, but can they actually efficiently secure their resources? And in order to answer this question, I took 2 species from either end of that gradient, a shallow specialist, Chaetodon baronessa. It specializes on a few corals and has a shallow distribution, and a deep generalist, Chaetodon octofasciatus. It feeds on many coral genera and has a deeper distribution, to look at their ecology more closely. Now, what we expect from shallow water ecology, if you think of each of these dots as a coral genera, and the size of the dot tells us how important that genera is in the diet of this species. What they do is they form pairs and they form territories around their preferred coral genera. They go around their territories, protecting their resource from specifics, and one of them feeds while the other one keeps an eye out for potential competitors. Now, we know in shallow water, when you lose coral habitat, you move away from that optimal ecology, and individuals and pairs increase the size of their territories. They feed on a slightly broader range of coral resources, and they spend more time moving around these larger territories and actually less time together as they try to circumnavigate those territories and protect them from others. So because of a steep decline in their preferred resources with depth, we expected this shift from optimal to suboptimal ecology in the butterfly fishes as you move into deeper depths. For our deep generalists, we kind of expected a similar ecology along the depth gradient. So what I did is I tagged a whole lot of fish, 39 Chaetodon baronessa pairs and 20 Chaetodon octofasciatus pairs. I followed them around for months on end, recorded their territory sizes, the density of prey within their territories, and the number of bordering conspecifics. And here's just an example of what some of those territories look like with their key resources outlined in lighter blue. You can see them stacked up along the depth gradient. The next thing I did was look at the behavioral parameters of these territories, so rates of territorial interactions, movement rates, how far they were moving in a given observation, how often they were paired. Now, the expectation is that deep residence should require more space because there's fewer resources at depth. And that's definitely the case. So you can see for the shallow specialist, their territory sizes increase with increasing depth, but not so for the deep generalist. The next expectation is that resource density within the territories should also decline with depth, but that wasn't the case. it seemed to be that deep residents were putting their territories on areas that had similar densities to the shallow territories, and in some cases even slightly higher. The next expectation then was that the total secured resources should be equal or decline with depth. So you have fewer resources in your territory, but your territory is expanding to try and get equal resources. But that wasn't the case because we didn't have a decline in resources within territories with depth, but we did have greater territories. The deep reef residents, particularly in the shallow specialists, actually had more secured resources, both in hard coral and in their preferred coral acropora at deeper depths than their shallow counterparts. So that's really great news for those deep fish there. The next thing we wanted to look at is what were some of the behavioral components. Did neighbor density decrease with depth? Did defense effort increase or decrease with depth? Did they have to move further for a given time period to get to their resources? And did they spend less time together? And you can see that neighbor density actually decreased with depth for our shallow specialist here in red. The effort that they had to put into defending their territories also decreased with depth. There was no significant change, but slight decrease with depth in the distance that they moved per unit of time around their territories, and there was no change in their pairing ratios. So it looks as though the deep residents here are actually doing quite well with a reasonably stable ecology. The next question I wanted to ask was, do they have depth-related diet changes, and do those diet changes potentially incur sublethal costs? So within the same territories, I followed the fish around, and actually in many other hundreds of observations as well, and I counted the number of bites each fish took in a 3-minute observation period, recorded the coral genus that they fed on, and the depth at the start and the end of the observation. And you can see that for both of these species, there's no change in the number of bites that they took from corals at any position along the depth gradient. The next question I want to ask, well, do specialists stay specialists and generalists stay specialists, or do they change what they're feeding on along the depth gradient? So the hypothesis here is that dietary breadth should increase with depth. And we have our shallow specialist, and you can see that they stay specialized. So their overall dietary niche throughout the depth gradient is very narrow. This varies between 0 and 1, and it's close to 0 at 0.07. And the deep generalist, in comparison, has this dietary niche of 0.23, so quite a bit broader. And if you look at these plots, each of the bars represents each of the colors represents a different coral genus, and you're moving along the depth gradient here. And you can see that there was high overlap in the diet of Chaetodon baronessa. They fed on 18 genera overall, and you can see there's a 95% overlap in what they were feeding on, and it's mostly acropora in this dark blue bar. In contrast, the generalist stayed generalist. It had little overlap between its diet at different depths, and you can see that it fed on twice as many genera, and it seemed to change what it was feeding on according to its availability and accessibility at different depths. So the specialist stayed specialist, and the generalist acted as a generalist. And with this decrease in acropora, actually what was really interesting is that at these deeper depths, Chaetodon baronessa was selectively feeding on acropora 40 times more than you'd expect, given its availability in the environment. So it was really seeking out these acropora colonies. So the next thing I wanted to know is the energy content of these acropora colonies similar along the depth gradients? So what I did is I extracted the lipids of two popular morphologies of acropora, tabular and hispidose acropora, extracted the lipids in the lab and looked at that along the depth gradient. And for both of these morphologies, there was no change in the percent weight of lipids that they had available as energy along the depth gradient. So it looks as though those acropora corals that they're feeding on in deeper water actually are providing reasonable nutrition as well. The ultimate test, though, is to see, obviously, what condition the fish are in deep water. So we recorded the length of the fish, the relative mass of the fish, the reproductive energy or the relative size of the gonads of the fish, and energy stores within the fish. And so if you take a histological slide of the liver of the fish, they can't store excess fats around their muscles so well, so they store in their livers, and you can count these white fat vacuoles, and more fat means better condition. And for our deep generalist, their condition didn't change at all along the depth gradient, and we weren't too surprised by that. What we really wanted to know is whether our shallow specialist had lower condition at deeper depths. And actually, this wasn't the case either. So it seems as though there's no change in total length, no change in mass, no change in the gonosomatic index of the females, and no change in the storage, energy storage within the fish in the males. And so this means that there's strong refuge potential in the ecology of these fishes, where the deep reef residents have more total resources than their shallow counterparts. They have less competition, they're having fewer competitive interactions. They're feeding on their preferred coral resources just as much in deep water as they are in shallow water. And they are supposedly getting as much nutrition from those deep water corals in the deep water as they are in the shallow water. But of course, the real test is, are there actually disturbance impacts and resilience shown on the deep reef? Are there differential impacts in shallow and deep reefs? And so the last stuff I'm going to show you today is 2 case studies looking at whether deep reef assemblages are more resilient to coral losses. Now, the first case study is from working with the California Academy of Sciences, the Rebreather team there. They were in Marinha in 2019, just after a high sea surface temperature anomaly. The anomaly peaked in late April when they were there in mid-May. So they were there when the bleaching occurred. So what they did is they recorded attenuation and bleaching along the depth gradient and the feeding preferences of 3 butterfly fish species on bleached and unbleached corals. So what you can see, if you look at the resource availability at the 2 sites that they looked at, you can see that the total coral cover decreased with depth, and that's cover regardless of bleaching impact. If you look at the corals that remained pigmented or did not bleach, you can see that actually the cover of pigmented corals increases with depth. So even though you have more corals in shallow water, you have more healthy corals in deep water. And if you look at the proportion of colonies that bleached or remained pigmented in 2 resource genera for corallivores, Acropora and Posolopora, you can see that the incidence of bleaching declined dramatically once you hit a depth threshold for Acropora, and seemed to decline quite steadily for Posolopora. So you've got more non-bleached resources for corallivores at deeper depths. So we wanted to look at the feeding preferences of the corallivores. Are they selectively feeding on healthy corals and avoiding majorly bleached and dead corals? And if you look at the y-axis here, what you've got is selectivity. Above 1 means that they're feeding on the resource more than you expect, given its availability in the environment. And you can see that they're selectively feeding on healthy corals, all 3 of these species. And you can see that all 3 of the species are selectively avoiding feeding on bleached and majorly dead corals, which is kind of what we expected. And of course, what we wanted to know is whether these selectivity indices are maintained throughout the depth gradient. And so what you here are looking at depth bins from 0 to 42 meters. And you can see that Ornithismus and Reticulatus, they continue to feed preferentially on healthy corals as you move through the depth gradient and avoid feeding on bleached and dead corals. But for Chaetodon palawensis, you get this interesting depth interaction where it seems as though they weren't able to avoid feeding on majorly bleached corals in the shallow water, whereas they were in the deep water, and vice versa. They seem to not feed on healthy corals as much as you'd expect in the shallow water, but fed on them preferentially in deep water, similarly to these other two species. And if you put the resource availability and the feeding preferences together and look at the actual total proportion of bites that were achieved on bleached and non-bleached corals through the depth gradient, you get this interesting interaction where all species were feeding more on healthy corals than majorly bleached corals in deep water, but not in shallow water necessarily, where a couple of the species were having to feed more on these less preferred majorly bleached corals in shallow water, and were not having to feed on them at all in deeper water. And so that looks as though there might be increasing opportunities for these deep reef residents going into the future. And what we expect is happening here is that depth is influencing both the bleaching prevalence as well as the conspecific densities, or the densities of competitors, is decreasing with depth. And so therefore, the number of bites on bleached corals is an interaction of these two ecological processes. The second case study that I wanted to show you just quickly in wrapping up is again in Kimby Bay in Papua New Guinea. And what you're looking at here is the cover of Acropora in the shallowest 10 meters of the depth gradient in 2013, and below that the density of Chaetodon baronessa, this strongly Acropora preferring species. In 2014, there was a crown of thorns outbreak. The crown of thorns fed predominantly on Acropora and diminished the Acropora population in shallow water by 2015. And we saw a similar collapse in the Chaetodon baronessa population, which is something that we generally expect in shallow water. It's not much surprise. Of course, what we wanted to know is what was happening below 10 meters. And I had some data from 2013 that suggested that there were potentially similar densities at these deeper depths. So I went back and sampled in greater resolution in 2015, and you can see that there seems to be this refuge effect where you have lost the shallow water population, but the deep water population has at least maintained itself and potentially increased. Now, we don't know whether these individuals or populations were already resident on the deep reef because we didn't have such great data from 2013, or whether individuals had moved down from these shallower depths. So I set up a quick experiment, and it's the last thing I'm going to show you now, where I set up 3 plots on another reef between 0 and 30 meters, 2 experimental plots where I moved all of the conspecific fish between 5 and 30 meters, and 1 control plot where I didn't remove any of the conspecific fish. I waited for 8 months whilst there was a crown of thorns outbreak, and we lost a lot of the coral in the shallow water, not so much in the deep water. And these fish in the shallow water were tagged, so I knew where the individuals were. And when I went back 8 months later, in the first plot, we'd had 1 pair migrate down to 18 meters, and 1 pair had disappeared. In the second plot, pretty much the same thing happened. 1 pair had moved down to 21 meters and set up on a plate, an Acropora plate there, and 1 pair had disappeared. And the control plot, we had many fish disappearing, and we had no record of species migrating down. So there's some evidence for vertical migration away from degrading shallow water habitats and possible effects of prior residents interacting with this. So the conclusions of these studies is that many species have broad depth distributions and deep distributions, though 25% of the species that I've looked at are limited to shallow waters, and the shallowest 5 meters. Coral diet specialists have broad and deep distributions, and specialists are also abundant in the system. However, the most specialized species are strongly biased towards shallow water. The deep reef residents have greater total secured resources at depths with no extra cost and effort in maintaining those resources. There seems to be stable feeding behavior amongst the specialists. The generalists are more varied. And the nutrition and body condition are stable through the depth gradient. There's potentially altered energy pathways in substitution, which I didn't talk about here, but we could talk about later if people are interested, or you can see it in the paper that I presented the information from. And more importantly, or most importantly, we have spatially differential impacts and disturbance outcomes, where deep reef provided habitat with preferred resources and a refuge for the deep reef residents. So the implications of this is that areas of deep coral habitat are likely not a panacea, they're not a cure-all for ongoing reef degradation, but they're potentially important refuges, at least for regional persistence, for some species of highly vulnerable reef fish, but only if deep water habitats do remain stable. And just a couple of bonus thoughts. I expect that we may soon observe resource pulses for coral specialists that are reliant on corals in shallow waters, and that coral reef futures are unlikely to be spatially uniform, and this includes along depth gradients. And just in summary, I just wanted to say that thanks to a whole lot of people, none of this research happens in a vacuum, and there's many more people even than are on this slide that have helped out with the research that I presented to you today. All right, thanks very much, Juan. That's the end of my presentation. So if you'd like to open up for questions, that'd be great.
Hi again. Thanks, Chancey, for your excellent presentation and you abording different topics about the importance for the deep and distribution for the reef fishes in general. In this moment I talk in Portuguese, only try to motive.
No problem. I apologize for not having Portuguese in my repertoire to join you.
Okay, maybe next time you talking about in Portuguese when you visit Brazil. Yeah. Ok, eu quero abrir então o espaço para as pessoas que ainda estão nos assistindo para fazer algumas perguntas sobre a palestra do Chance. Suas perguntas podem ser em português ou em inglês. Eu vou tentar, as perguntas em português, tentar de traduzir para o Chance, e as que estão em inglês, eu acho que o Chance também poderia ter acesso direto para ler, em caso ele não consiga entender meu inglês. Então, enquanto as pessoas vão se animando, eu vou aproveitar e fazer algumas perguntas aqui para o Chance. Chance, I wait for the question for the different people try to write in this moment, but I try to formulate one question with you about your presentation. For example, it's more one curiosity because I remember the article for the Luis Rocha explaining the reef is not one refuge because you have different reef fauna in this place. And your article, maybe you propose different migration in different reef, the shallow water for deep water, but I don't sure is one capacity for the resist for different climatic change. For example, what are your opinion about this point? Because probably this is more specific for one specific family, for example, cattle and tedium, but I don't know, is this case, for example, for top predators or another families using more another resource?
Yes. Yeah, I think there's a number of factors that play into this. So one of them is where you draw the line between a shallow reef and a deep reef. So we definitely see a faunal break or a number of faunal breaks at about 60 meters depth, where you have very few species in common in the shallowest 30 meters. And then once you get below 60 meters, you have very few species in common. And so that's kind of generally asking whether mesophotic reefs might be a refuge for shallow reefs. The work that I do is kind of more limited within what we would consider the shallow and upper mesophotic depths. And I think most researchers would agree that if there was to be a refuge for shallow reef communities, it would be in the upper mesophotic depths, where you do have some overlap between these two communities still. And the second thing is that people who have made statements previously, whether deep reefs are a refuge or not, have tended to be on a yes, no kind of basis. So the idea has been more looking to see whether deep reefs are actually untouched. Do they have any impacts on that the shallow reefs experience, or do they have no impacts that shallow reefs experience? So what my work has tried to do is try to look a little more detailed at the ecology of reef organisms as you move along the depth gradient and get a more nuanced answer to that question.
Okay. Okay. I wait for another question here. The people
Yeah,
send.
no problem.
Yeah.
It's just before Christmas, so I wouldn't be surprised if everyone is off enjoying
Yeah.
themselves already.
Yeah. Probably the people assist after your record because all the time is this gravation record is
For sure.
in the
Uh,
YouTube
and you
link.
can see in my second to last slide, I did leave my email address there. So if anyone has any questions after
Yeah.
this live presentation, they're welcome to
Yeah. Perfect. Maybe you can write in the
in the
YouTube,
comments.
or you can send me, and after I can put here
Okay,
your
that
email.
sounds
We put your email here.
yeah,
Uh, well, I have another question. Sorry.
yeah,
Uh,
no problem.
you see difference for the abundance in different gradients for the diff, and your plots, for example, you see move different species for the shallow for the deep areas,
Uh-huh.
but you see changing the abundance and the biomass, or only you see it from the species richness.
Uh, no, you see changes in the abundance as well as species richness as you move along the depth gradients in general. Um, and then with the experimental plot where I had recorded some individuals moving deeper onto the reef when there is an availability of resources, we expect that that might be have prior residency effects, so that deeper reefs probably have lower abundances than shallow reefs. And if you already have individuals occupying those few habitats that are available, then shallow individuals won't be able to move down. But if those habitats are unoccupied, then shallow individuals would be able to move down onto the deeper reef.
Mhm. Hmm. Ok, perfect. Pessoal, alguma outra pergunta para o Chance? Senão vai ficar um monólogo eu falando só com ele. Quem tivesse alguma dúvida pode escrever aqui, pessoal. Fiquem à vontade. Ainda temos 5 minutos para fazer algumas perguntas para o Chance aqui. People today is very nervous.
Yeah, that's no problem. If there's no questions, we can wrap it up and ask some other time. I had a question for you though, Juan. I know a number of your the members of the group that you work with latitudinal gradients and changes in reef fish ecology with latitudinal gradients. Are there any general findings in relation to how species distributions and ecologies might change at different points of latitude going into the future from your group?
Yeah, probably I suspect this change in distribution for the species when the latitude, because the climatic change, the species migrate for the subtropical areas, and probably you have the tropicalization case, for example. Different wars in Australia and Japan show this migration, because you have the migration, horizontal migration, but you observe the vertical migration in your case. But I think
Mm-hmm.
the both migration is very common for the refuge because it's one three-dimensional space. And well, I think this is very important. Try to understand the vertical and horizontal migration for the future.
Yeah, yeah. One of the benefits that horizontal migration has over vertical migration at this stage in terms of studies is that we have a lot of historical data or long-term data at different latitudes. So we can see that quite easily, the species that move further south as climate warms up. With the deep reef environment, we have few studies
Yeah,
that have strong abundance data and species distribution data, long extensive depth gradients, maybe 0 to 15 meters perhaps, but very rarely beyond that. And I think that's something that's going to be important going into the future as more and more groups, like I know you're about to join a group that's going to work on deep reef ecology in Brazil, which is really awesome. And as we get more and more of these groups studying deeper reefs, having this baseline data set to be able to then do long-term monitoring is going
yeah,
to reveal some really interesting patterns going into the future, I think.
yeah, yeah. I think this is the new frontiers is more accessible in this moment because you winner more methods for sampling these different gradients and is
Mm-hmm.
no more calls for example for Brazil or Colombia and different countries in the development. Okay. I have one question here for Carlos Eduardo. Ah, how do you see the problem the deep reef with good refugia in terms of the distance they cause? Generally, when the deep reef is far from the coast, they receive less impact. Do you
Okay.
agree?
Yes, well, more isolated reefs tend to have less of the impacts from land. So, for example, increased nutrient loads or increased sedimentation from increased rainfall, and those sorts of things. I The increased water flow that might occur around some isolated environments might also mean that they have less susceptibility to coral bleaching, but I don't think that has played out going from what we've seen in the last few years where there's been global coral bleaching events. But it's an interesting question that you raise, and it's actually a question that I studied in the first paper I released, the 2016 MEPS paper, which looked at the distribution changes not just along depth gradients, but also with distance from shore. And there was definitely an interaction that the further from shore you were, the clearer the water was, the more abundant corals were at deeper depths. And you saw a slight shift in the depth distributions of quite a few coral-associated species, where they might have a limited distribution in the shallow water, close to the land, and then as you move further away from land, the distribution got broader and broader and deeper. So there are definitely interactions between distance from land and depth that we've seen. So it's a great question. Thanks, Carlos, for asking that.
Pessoal, alguma outra pergunta pro? Ainda temos 2 minutos para uma última pergunta para o Chance. E eu coloquei o e-mail dele aqui. Quem quiser assistir depois ou compartilhar o link para depois enviar alguma pergunta sobre a apresentação dele ou sobre alguma de suas pesquisas atuais, o e-mail do Chance está disponível aqui no chat do canal do CEBIMar.
I just remembered a couple of things I could say about that as well, while we're waiting for potential other questions to come in. So I've done a lot of extensive offshore trips for many months at a time. So, for example, in the Coral Sea, close to the border of the Australian Economic Zone, I've spent a month out there diving. And surprisingly, some of the reefs out in the Coral Sea had quite low coral cover in the shallow waters, even around about 15, 10, 20% coral cover. So it definitely seems as though bleaching impacts and storm impacts do affect some of those more isolated reefs also. one of the other things I was thinking about, Juan, we were talking about accessibility and how increasing accessibility is changing the way that we can study these deep reefs. So all of the work that I presented today, that I collected the data for myself, was with nitrox and twin tank systems. So you're
Yeah.
spending, maybe 10 or 15 minutes at these depths, being able to record what's happening in the deeper parts of the reefs, and then you're doing as much as you can on the way up with all these other ones. One of the great things about joining the California Academy of Sciences is that we were planning to have myself be trained in rebreather diving, and we haven't been able to do that yet because of COVID, but an extension of my contract means next year we'll be able to do that. And some of the work that I did took months and months and months on scuba with nitrox, and it could be achieved in just 2 or 3 dives on rebreather technology. Being able to spend extended period of time, 50, 60 minutes in one place. So that would be really interesting.
Good. Okay, have you new question here for the Luciano. He's starting watching the presentation, but it's very interesting. Have you noticed any difference in the last few years called associated climatic change, these gradients?
Yes, well, I guess, the most easily visible impact of climate change is on coral reefs is the bleaching of corals. Um, and definitely we're seeing that there's an attenuation of that with depth, so that you have generally more bleaching impacts in shallow water than deep water. But in some cases, for example, in the Chagos Islands, there was a report released last year that showed, following for 30 years, actually, that in the last couple of years, there's been strong bleaching impacts even at 30 meters depth there. And there's almost no coral cover left on those reefs after 30 years of following them, from about 80% coral cover down to about 5 or 10% coral cover now. The deep reefs were resilient for a long time, and then just in the last couple of years, they've degraded also. And then Luis Roja, or Roja, I should pronounce that properly considering I'm speaking to the Brazilians. He and his group have a science paper out in 2018 that shows that there's definitely a lot of anthropogenic effects on mesophotic coral reefs. So whether that be sedimentation from storm events, where you get the sediment and rubble getting pushed off of shallow reefs and down onto deep reefs and smothering deep reef corals, or whether it be fishing equipment or just general trash discarded from boats passing by, or fishermen, there's definitely impacts on those deeper reefs.
You have another question here for Carlos. Most the butterfly you have studied are coral feather. What you expecting for generalist butterfly could use the deep reef of the refuge too?
Yeah, so the reason why I concentrated on the obligate coral feeders is because they have the highest losses in shallow waters when we have coral loss. So, for example, Ketodon baronessa, that I focused a lot on as the shallow specialist, is one of 2 species that have shown really high abundance losses when we have coral loss in shallow water. So what we expect is the more generalists you are, the more number of niches you can occupy successfully. And so that means that you should be able to more successfully move along the depth gradient and occupy those changing niches more easily than the coral specialists. So I'd expect that for generalist butterfly fishers, they probably have just as good, if not a better opportunity to utilize deep reef environments, assuming that the plankton that they feed on, if they're planktivores, it's going to be reasonably evenly distributed along broad depth gradients. At some point, the light attenuation is going to stop them being able to see the plankton very easily to feed on it. And we don't know much about invertebrate communities, or mobile invertebrate communities, worms and crabs and things like that, and how they change along the depth gradient. But the assumption is that you can, we don't know much whether generalist butterfly fish, for example, that feed sometimes on corals and sometimes on invertebrates, only ever eat one type of invertebrate, or if they eat any invertebrate that's there. So we don't know enough to know whether there's specialization within those groups. But the general principle would suggest that they would do just as well, if not better, on deeper reefs.
Perfect. Well, Chancy, again, thanks for accepting your talk about your excellent work. And I finished the session for the question because it's the time.
Yep.
E, pessoal, muito obrigado a todos que estão presentes, que estão assistindo e aproveitaram para perguntar algumas coisas para o Chancy. Eu quero desejar para todos umas felizes festas e em família e boa saúde para todos para o próximo ano. E a gente fecha nossos seminários por esse ano. Começaremos de novo com a sessão de palestras depois do dia 10 de janeiro. Agradeço a todos os participantes e thanks again, Chance, for your participation.
No problem, Juan. It's always a pleasure to be able to share the passions that we have, and it's very generous of you to invite me along to your scientific community. It's really a pleasure to share the research that I've done with them. Thank you.
Ok. Pessoal, muito obrigado e até o próximo ano.
Ciao.